This method can help answer key questions in the fields of immunology and chronobiology, such as how microbial components alter the molecular clock. The main advantages of this technique are that it is easy to perform, the effects of different microbial components on the molecular clock are assessed, and it is highly reproducible.
To begin the protocol, prepare culture medium by adding fetal bovine serum to rPMI 1640 to a final concentration of 10%. Prepare 10 milliliters of challenge medium for each of the PAMPs to be tested in 50 milliliter tubes by adding the listed PAMP culture medium. Spray a previously prepared mouse trunk with 70% ethanol and wipe it with a paper towel. Place the mouse on its back, slightly tilted onto its right side. Next, cut away the fur using dissecting scissors along the mouse's left side, about halfway between the front and back legs. Using forceps, grab the peritoneum and carefully make an incision without damaging the spleen. Remove the spleen with forceps and place it into a sterile 50 milliliter tube containing approximately 10 milliliters of culture media on ice. Then transfer the spleen with 2 milliliters of culture medium to a small sterile petri dish. Homogenize the spleen by grinding it between the frosted portion of 2 sterile frosted slides. Once thoroughly homogenized, pipette the 2 milliliters of culture medium containing the splenocytes through a 40 micron nylon cell strainer into a 50 milliliter tube. Add 8 milliliters of cold culture medium to each of the 50 milliliter tubes containing the splenocytes for a total volume of 10 milliliters per tube.